Well, thank you, Dr. Hema, for inviting us to this virtual conference on cancer. This is the first time we are doing a virtual conference, and I'm so happy to participate in this. We wish Crescent University and your team good luck in conducting this conference, and then you're doing an excellent job, and we are so happy to participate in this. For the next 15 to 20 minutes, I'll be presenting about an important part of cancer, that is finding a therapy for a drug resistance cancer. And before I go further, I would like to introduce myself to the other audience. I'm Kannan, and I'm a professor in Department of Radiology and Bioengineering at University of Missouri, Columbia. And I run a group mainly focused on cancer diagnostics, cancer imaging, and cancer therapy. The topic that I'll be dealing with today is AXL targeted lung cancer therapy. AXL is a biomarker that recently been established to be overexpressed in lung cancer, kind of like too much in the lung cancer when it becomes resistance. So AXL, accelerative targeted lung cancer therapy, AXL being a biomarker. This particular paper, it appeared in Nanomedicine last year, the month of August, and I want to summarize the data that we presented. Just regular statistics about cancer in the year of 2019, over 1.7 million people got affected by cancer, and then over 600,000 people were dead because of cancer. Just to give it in a different perspective, there were close to 4,800 cases every day, and close to 1,600 people were dead because of this cancer. These numbers looks very different when we compare with the real COVID-19 situation now, but this is equally bad. The lung cancer statistics for the year of 2019, there are close to 250,000 people were diagnosed with lung cancer, and over 145,000 people were dead because of this cancer. You can see a very sharp increase in the number of cases as well as the number of deaths here as the proportion goes up, mainly because the cancer as such, the lung cancer is a deadliest cancer known in men and women. I think second deadliest cancer that is known in men and women. And the therapy for lung cancer is usually like it works very well initially, but after some time the cancer comes back with a vengeance and it becomes resistant to the existing drug. Let us see what is happening in the real clinical situation. A patient comes to the doctor, and then after the patient is suspected with lung cancer, they take a small biopsy that you can see from the red place, where they take a small amount of a tissue and then do the biopsy. They confirm that like it is a lung cancer. After they determine what is the molecular signature that is present in the cancer, for example in the case of EGFR mutation, we go with something called a tyrosine kinase inhibitors. After with tyrosine kinase inhibitors, now there is a third generation tyrosine kinase inhibitor called osimertinib. If you treat with that, 1 year, 1.5 years, the patient will be like a symptoms free, patient will be like survive with no disease at all. But exactly after 14 to 17 months, in the case of osimertinib, it is a little bit more than that, it is like around 20 months or so, the drug stops working and there is no other drug to replace that. Okay. The central part is like when this drug stops working, the cancer starts coming back again, and it ramps up so much, it gets metastasized, and it actually succumbs the patient very bad. Given in a little bit of cartoonic way, this is a drug. What you are seeing in the left side of the screen, you can see the drug. This is a lung cancer. Once it becomes resistant, you can see that the cancer pushes it away. It removes the drug from the place, and then it goes over the. It kind of like removes from the tumor, and if there is no drug, you cannot treat the patient. There are various markers that are being known, that are being identified to show that has prominence in the case of lung cancer. There is a separate mutation called T790M mutation, which has been shown to become for mostly expressed in the patient with drug resistance. AXL is one kind of a class. Okay, AXL is being overexpressed in patients. Around 20 to 25% of EGFR mutant patients, and that is the reason for the resistance. So, you know the cause for the resistance. Can we find the therapy? That is the whole idea of this particular talk. But do we know that there is a cause? This is a statistics which we did in our lab, based on tumor genomic data. The red line that you're seeing down, these are all the patients overexpressing AXL. Patient overexpressing AXL shows low month survival and have a lower months with the disease free, which means they are very soon succumb to the resistance and very soon they die. So AXL, in our opinion, it is participating in multiple components within the cell, and there is a strong literature behind it. AXL directly gets involved in the proliferation, and the AXL gene, when it comes out from the nucleus, AXL mRNA, which kind of translates and then forms into a AXL receptor, which kind of overexpressed in the tumor cell. Overexpression of the tumor AXL is directly relates to the AXL mRNA. Can we stop this AXL mRNA from the production? If the AXL mRNA is stopped, which means the overexpression of AXL can be stopped. If the AXL overexpression can be stopped, then our traditional tyrosine kinase inhibitor, which works on EGF receptor, where you can see the erlotinib, it's on the other side of the slide, will work. So the central piece is if I can knock off AXL, or if I can downregulate AXL, if I can stop the AXL overexpression, then the tumor would start responding to the existing drug. We will not go for any animal data today, but we will tell you how we designed our nanoparticle for treating AXL in this particular talk. How we wanted to work, how we wanted to move further on this therapy. We wanted an antibody to target the cell so that the lung cancer can be specifically targeted. The lung cancer can be specifically targeted using EGF receptors, which are overexpressed in the cancer, and we wanted something which is biodegradable, a nanoparticle which can degrade within the body so that it doesn't give any toxicity. And what we wanted to do here is something called an siRNA, the silencing RNA. This silencing RNA targets that AXL mRNA. This is the number 3 in the slide that you would see, the siRNA, which target this oncogene, which is causing the resistance. In this case, it's an AXL, will knock down this particular AXL mRNA. So, what is the ensemble? We wanted to develop a nanoparticle which should be biodegradable, which should be attached with an antibody so that it can find the cancer, and we wanted to attach that with an siRNA so that the siRNA can get inside the cell and knock down the AXL mRNA. This is the whole idea and the whole story behind how we have went ahead with this in a nutshell. The other piece of information that I would like to bring it here, the challenges of siRNA. siRNA is the therapeutic, so to call, the molecule here. The siRNA as such is very unstable. It gets decomposed within 10 to 15 minutes or sometimes even less than 1 or 2 minutes in blood. How we are going to deliver that siRNA into the tumor? Can we handle it? There is another reason why we chose nanoparticle for this investigation. So this is a molecule which we designed way back in 2016 and it was published there. This is a unique structure. It has a gelatin molecule. Gelatin is a protein. Gelatinase, this particular one is made out of swine. The gelatin nanoparticle is attached with the cetuximab, an antibody, which targets EGF receptors. EGF receptors are overexpressed in most of the non-small lung cancer, and then AXL siRNA is attached in between the cetuximab and gelatin. You can see that in the inlet here, insert here, the siRNA is kind of protected between the antibody and gelatin. If it is protected between these two, the serum nucleases or the proteins will not degrade this siRNA. You are kind of protecting it. So this was a central piece. We published it. We extended the same concept here. So we attached the AXL siRNA to gelatin nanoparticle, which is surface targeted to EGF receptors using cetuximab. Just to show you, these are all perfect sized nanoparticle. What we said, GNP is a gelatin nanoparticle which is attached with an antibody. You can see that it's spherical, and when it's attached with AXL siRNA, you can still see the spherical. It's kind of 180 to 200 nanometer. We have an extensive characterization, stability studies, and decomposition. Everything we studied, and we listed that in the paper if you're interested to go through. But there are few results which I would like to highlight today. What is the first thing that you wanted to see? I made this nanoparticle which is targeted towards the cancer cell. But crucial part is, can it take the siRNA inside the cell? If it is not taking the siRNA inside the cell, it is not going to be useful. We did a small experiment wherein we attached this siRNA with the fluorescent label. In this case it is CY5. We attached the AXL with the CY5. We made the same nanoparticle, and you can see that like free siRNA with CY5, we took the cancer cells, we treated with the DAPI, a nuclear dye, that untreated cell with that blue dot, which means that like it is a cancer cell only with the blue dye. And when I treat that with gelatin antibody, you can see no color change. When I treat it with siRNA with CY5, there is a siRNA is attached with fluorescent label. Still, I don't find any uptake within the cell. These are all the fluorescence microscopy images. Now you see the fourth quadrant that we have, where we said like AXL CY5, wherein you can see the CY5 concentration close to the or in the cytoplasm. It is not overlapping with the nucleus, it is close to the nucleus. The blue is the nucleus and the red color is from the CY5. So what we have proven from this experiment is that like we were able to transport siRNA from outside inside the cell. And another question which we always ask is like, now you know that like it is transporting, is that stable enough? This experiment is kind of like is a two-part, I would say. What you are seeing on the left-hand side is unbound siRNA, where you are seeing a very strong band. When you add siRNA as such to this page, everything gets decomposed. You can see like even in 0 hours and 0.5 hours and 1 hour, you see a strong band where it is correspond to the smaller pieces of siRNA. On the right hand side, we spotted nanoparticle, wherein the nanoparticle siRNA is bound. Even up to 48 hours, you see a strong band on the top, okay, which we call it as a conjugated siRNA. Very strong, present there, and no decomposition. You don't see any band at the bottom. Confirms that the siRNA is intact with this nanoparticle. So what we have shown, siRNA is intact with the nanoparticle. Nanoparticle can transport siRNA inside the cell. So now we'll go a step forward here. There are 3 pieces here. I would go with A, B, and C. I would like to you to focus only on A for a second. What we are trying to do here, we are going to stop the AXL mRNA from production, which we saw that in the cancer cell. And this AXL mRNA should not allow the expression of AXL. So you should not see AXL at all. So we took cancer cell, we just treated that with siAXL, okay? That is siRNA of AXL. And what you are saying is an NM is being nanomolar. Okay, the band is in the western blot. You can see at 0, which means like when you don't have any siRNA, you can see a very dark band of AXL, actin being a regular control. But as we increase the concentration of AXL, you can see that like after it crosses 25 and 250 nanomolar, then the concentration of AXL goes up. If the concentration of the AXL goes up, it's kind of like it brings down the AXL mRNA substantially down and completely knocks down the AXL from the picture, from the cell. So this is a good data that we got, and we wanted this to be repeated with the nanoparticle. Okay, now I want your attention for section B here. What you are seeing is a GAP SA-AXL at 6 hours. That is like after you treat 6 hours, the concentration is what we predetermined with the AXL, but this is with nanoparticle now, right? The nanoparticle is the one that is going to work because plain siRNA is going to disintegrate in the body. GAP SA-AXL at 6 hours, and even if you treat it with 3 hours, you can see a substantial decrease of AXL protein production. When you have an erlotinib added to it, you can see the AXL disintegration is completely gone. So AXL production is completely gone. Just to put it in a quantitative way, I want your attention now in the C part of the figure. What you are seeing in the y-axis is an AXL downregulation in percentage. In the case of SA-AXL, it was around 80%. In the case of GAP SA-AXL, it was around 75 to 78%. In the case of GAP AXL plus erlotinib, it is similar to that. But what is that important is erlotinib alone is not doing anything, and only the AXL is being controlled by the AXL mRNA. Yeah, what we have seen so far is AXL is being downregulated by using the nanoparticle, and by using AXL siRNA, we were able to deliver siRNA inside the cell. But that is not our end goal. What is our end goal? Our end goal is to sensitize the existing drug. So we took a couple of cell lines. One is H820 and H1970. When we treated with erlotinib as such, you can see that IC50, which is in the black color. I want you to focus on the A side of the picture. You can see that the IC50 were 10 and 22 or 23. But when I knock down AXL, you can see that like AXL with erlotinib, the IC50 was substantially decreased. In the case of 820, it came from 12 to 8. In the case of 22 to 15 or 16 or something. So there is a decrease. It is not only pure erlotinib. When you knock down the AXL using simple siRNA, you are able to bring down the IC50 value. Now let us focus on B. B contains the nanoparticle with siRNA, and what I am saying here is the black color at the end with H820. You can see that like now, which was 10 earlier, now it is close to 5 or 6. It is a substantial decrease when you put GAP SA-AXL plus erlotinib. There is a decrease. So you are able to sensitize the cell for erlotinib. So what we have done is like we were able to find out the main cause for the resistance. We were able to pick up that siRNA attached with the nanoparticle. We investigated whether the nanoparticle is stable, whether it can take it into the cell, whether it can bring down the AXL protein, now whether it can also sensitize the cells. We were able to see the whole gamut. We also established that in animals, but it will be published in the future part. But what is interesting is that doing exactly the same thing what we are expecting or anticipating in the mechanism. For the next 3 or 4 slides, I will be focusing only on the proteins, how this AXL impacts, and present you the comprehensive mechanism of AXL mediated drug resistance. There's plenty of Western blot here, but I want your focus on A now. You can see the phospho-AKT levels, that is the third row from the top on the picture A. Phospho-AKT levels have gone down, which means like like AXL is working. All the other proteins, like if you are interested, like there's a lot of description presented in our paper, but it shows that phospho-AKT is a salient feature. Now you can see that like the phospho-EGF levels, when you treat it with along with erlotinib, is going down. Okay, so we were kind of like towards the mechanism. Now like it is sensitized. It also brought down the AXL with the Western blot, and then we are also showing that in phospho-AKT levels are going down. We have invested close to 40 to 50 different proteins, and we found out like there are crucial proteins and many molecules which act together are coming down in aiding the AXL to become resistance. But when we knock down AXL, all the other proteins dies down to make the erlotinib to or the TKI to work. The GAB AXL plus erlotinib, like you can see from the cell lines, it induces the apoptosis at 48 hours, which is pretty high in the case of our nanoparticle, and in the case of PCR data, the fold change for AXL has gone down, whereas the MCF1 and TP53 has gone up when you treat it with when you pull down with AXL, when you knock down with AXL. Just another experiment to another data informatics to identify whether there is an association between AXL and vimentin. AXL, vimentin, and encadrin. AXL is known for metastasis. Like it becomes drug resistance and it becomes metastasis. So by this, we were able to see that in multiple cell lines, there is a close association between the AXLs and vimentin. So there is something which is key, because the AXL is being kind of a main molecule in making the cancer cells to move from the center part to go on metastasis in other places and become more EMT. It happens after the epithelial and mesenchymal transition. So you can see that there is a strong association between the AXL and vimentin. So we became interested to see, when I knock down or when I bring the AXL protein down, what happens to the vimentin? So you can see from A that when I bring down AXL plus erlotinib, please focus on A on the first row, you can see that the vimentin levels are going down. Okay, a key marker for EMT, and also you can see that encadrin and in figure B is going down, the another key marker. So it kind of links, it kind of says that AXL is linked with the EMT. When I bring down the AXL, then the EMT marker substantially goes down. So it essentially says, by bringing down AXL, you can even stop metastasis. Or if you are able to bring down the AXL protein, you can work on the metastasis, or you can save the patient. Now you have this nanoparticle which is with siRNA, selectively knock down the AXL, which can be of huge help for the metastasis, or stopping the metastasis, I would say. So we did the CRISPR. I want you to focus only on the figure F here. We selected a particular area. We did the CRISPR to kind of substantiate the data that we got. And going further, we kind of investigated a lot of miRNA. And the interesting part was miRNA 92A, which is also reported in the literature, have a strong association with the AXL. When the AXL goes down, there is an implication with MIR92A. We have so far seen the various molecules which place in part with the AXL. The one that you are seeing here is the role of AXL with MMP9 and MMP12. What you are seeing in the x-axis here is this percentage of change after treatment with AXL siRNA. And what you are seeing in the x-axis is the various molecules. I want your focus towards GAPC-AXL. You can see that the MMP2 and MMP9 got significantly reduced. When you have put GAPC-AXL plus ER, you can see a close to 100% change of MMP2 and MMP9. MMP9, I would say, after you are treating with AXL. What you are seeing on the leftmost extreme is H820AK, which means 820 cell line after CRISPR knockout of AXL, which also shows that MMP2 shows a significant decrease after the AXL is being knocked out by CRISPR. Taken together, where are we now? I'm going to take this particular slide. This kind of summarizes the role of AXL and the reason for cancer resistance. What we have seen today is kind of a multiple pieces, but this kind of summarizes. First, we have shown that AXL is the cause or one of the causes for the drug resistance. Then we made a nanoparticle which can act as a therapy to suppress or knock down the AXL mRNA or AXL protein to sensitize the drug. Then the third part is finding the mechanism. So taken together, we were able to mix and kind of like put the puzzle together to find out what happens when you suppress AXL. When you suppress AXL, there is a changes in N-cadherin, MMP2 and MMP9, and which in turn has a role with Vimentin, and then we were able to find out there is a correlation with TGF-beta, PI3K, AKT, and how it affects the growth and proliferation and how it leads to an apoptosis. Overall, we were able to convince that, or we ourselves got convinced that AXL plays an intricate role in the drug resistance. One piece of small information here: role of AXL for the drug resistance is not limited only to lung cancer. It is also very well known in ovarian cancer, and it is being investigated every day in other cancers too. So this is an evolving target. We gave a highlight. We showed like what we are going after with this particular marker. Well, like I would like to summarize my today's talk with 4 important points. The first point is about the drug resistance. It is a major obstacle. There is no problem in developing a drug for treating cancer, but we should more focus about treating drug-resistant cancer. That is the first emphasis I would like to bring it to you. The second point is about what we have done in our work now. We have developed a nanoparticle which is attached with an EGFR antibody and an siRNA for treating AXL. And third is about the results that we have obtained so far. We have used drug-resistant cell in this particular work to show the efficacy of GAPC AXLs, which reduces mTOR and EMT signaling with concomitant increase in the p53 expression. And based on the molecular changes and the amount of AXL that is getting silenced, the cells get sensitized by TK by at least by 2-fold. In the mechanistic part, our results show that AXL expression has an important role in driving the TKI resistance and its close association with energy-dependent mitochondrial pathways. Taken together in one line, AXL is a crucial part for drug resistance. If you can suppress or knock down AXL, you can reverse the drug resistance, or you can bring the reinitiate the drug sensitivity. I would like to take this opportunity to thank everybody contributed to this project. Dr. Joshi, she is part of our bioinformatics team, and Dr. Upendran, who has been integral with our team and specialist in molecular biology and also developing small animal models. Dr. Hammer is a pathologist, a board certified pathologist from our university, who has helped us in determining the role of AXL. I also thank all the other current team members, Dr. Suresh, who is the main part of this work, who worked on this complete project, that is his project. The other members like Abhilash, Dr. Lee, and other team members are also being equally thanked. And thank you for listening to my talk. And this is a virtual conference, and I thoroughly enjoyed presenting this data to you. It's kind of a little challenge, but I totally enjoyed sharing my experience and sharing my research data with you. Thank you all.